Okay, welcome back. So we've talked about what membranes are made of. We've talked about what the fluid mosaic model means. We've talked about what it means to be selectively permeable. We've talked about how molecules can pass without using any energy, how they can pass across the membrane through two different ways. One is diffusion, which doesn't use a protein, and one is through a channel or a pore that will use a protein. So next, let's talk about movement of water. Sometimes you can't move the sugar across the cell membrane. You can't move the big solute across the cell membrane, but your cell still wants to get to a place of equilibrium. So instead of moving the stuff inside the water, it moves the water itself. And that's called osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. Water is going to move to where the salt is. So if you have a lot of salt on one side of the membrane, so here you have a lot of salt, and here you don't have as much, or sugar, whatever solid. Here you have a lot of sugar and not a lot of sugar on this side. So water is going to move to where the sugar is. It's going to follow the sugar. So if there's more sugar here, it's going to move that way. So you see the water moving across here and moving up. So instead of the sugar moving and getting a balance, it's the water moving and getting a balance. So whatever solid you had here, if you had a lot of salt on one side and not a lot of salt, the water would move over. If you had a lot of anything on one side and the stuff couldn't move, the water will move to get to a balance. Okay, so water diffuses from lower concentration and moves toward higher concentration. That is moving down the membrane, or that is moving down the concentration gradient for water. Even though it's against the gradient for the stuff, it is down the gradient for water. So this does not take energy because it is still moving down the water concentration. And it'll keep moving until the concentration is equal on both sides. So here the water just kept moving and moving until at this point, it looks like the little sugar molecules are about the same distance apart on both sides of them. You've got about an equal concentration. Okay, so here's a little clip from your textbook about osmosis. Oh.
The plasma membrane is permeable to water molecules, and the movement of water into and out of cells is critical to life. Diffusion of water molecules across a selectively permeable membrane is a special kind of passive transport called osmosis.
Okay. So this movement of water is going to play a lot of important roles in the survival and the function of a lot of cells. So it's very important that cells grow in areas where they have the right amount of sugars and salts around them so that they can stay in a balance with their environment. So tonicity is the ability of the surrounding solution to cause a cell to gain or lose water. So if the solution around a cell has a lot of extra salt or sugar, it's going to cause the water in the cell to move out to follow the salt or sugar. If the water outside of a cell does not have any salt or sugar, it's going to move into the cell, follow where the salt and sugar is into the cell, and cause a cell to blow up and have too much water in it. So it's very important that you keep those solid solutions matched for the cell to be able to grow. So the tonicity is going to depend on the concentration of stuff that can't cross the membrane, how much of that is in the cell and how much of that is out of the cell. Osmotic pressure is how much force is generated when pure water moves through a cell to an area of higher solute concentration. So if you put a cell in a cup of water that has a lot of salt in it, that's got a lot of osmotic pressure because that water is going to try to move out of the cell into the salt solution. That's what osmotic pressure is. The pressure, how much pressure there is for that water to try to do osmosis. So an isotonic solution is when the concentration of salt and sugar, the same solid concentration, is the same inside and outside. So there's not going to be net movement of water. Whatever moves into the cell is going to be balanced by what goes out of the cell. So if you are a nurse hanging an IV, you need to make sure that that IV is isotonic to that patient's red blood cells so that their cells stay the same. If you gave them a hypertonic solution, A hypertonic solution means that there's more salt and sugar out in that solution compared to the cell. So there's too much. It's hyper means high. So there's a high level of salts and sugars in your solution. In a hypertonic solution, your cell is going to shrivel because there's so much salt and sugar, so much solute around the cell that the water will move out of the cell and follow where the solute stuff is. So the cell will be shriveled. If you gave a patient a hypotonic IV, hypo means low. So hypo rhymes with low. So that's how I remember that. A hypotonic solution has less solute concentration. So something like pure water that doesn't have as much salt and sugar as your cell has. All that water is going to say there's more salt and sugar inside the cell. So it's going to move inside of the cell and keep moving inside of the cell and keep moving inside of the cell. And if that continues, your red blood cells will fill up with water to the point of just like if you overfill a water balloon, it just eventually pops. Your red blood cells are going to do the same. They're eventually going to pop. So if you are a nurse who wants your patient to get better, you are going to have to make sure that you give them something like normal saline that is isotonic. Okay, so please be able to tell me what happens to a red blood cell. In an isotonic solution, it's going to be healthy and functional. Salt and sugar, solute is balanced inside and out. In a hypertonic solution, there's a lot of salt and sugar outside of the cell compared to what's in. Water moves out of the cell and it shrivels. Be able to tell me that. In a hypotonic solution, that means there's a low level of salt and sugar outside compared to what's inside. So water follows the salt, water follows the sugar, it moves into the cell. The water will overfill that cell and the cell will lyse, it will burst. Okay? We'll talk in a moment about what happens to plant cells because their cell wall actually makes them a little bit immune to a certain level of osmotic pressure, so they behave a little differently. Right now, just know what is going to happen to your red blood cells in these different environments. Hypertonic and hypotonic conditions, those types of environments do exist on Earth, and so a lot of cells can't live in places that are very hypertonic or hypotonic. But some organisms have developed evolutionary ways that have allowed them to be able to function in hypertonic or hypotonic environments. One way is with rigid cell walls that will just provide a pressure that balances that osmotic pressure. But things without cell walls can also develop osmoregulation. Osmoregulation is the ability to control how much water is in your cell, to control the solute concentrations and the water balance so that you don't either shrivel or burst. So one example is this paramecium. This is a paramecium. It's a little unicellular eukaryote. It is hypertonic compared to its pond water where it lives. And so it actually has a contractile vacuole that acts as a pump. So that prevents water moving into or out of the cell because it can pump the water to keep that water balance inside of the cell at the concentration that the paramecium wants it. Plant cells have a cell membrane around them that help provide some balance. So instead of water just pushing it in and in, if you imagine that water balloon that was overfilling and bursting, but if you were holding it and keeping it in your hands, your fingers would actually push back, provide that pressure that pushes some of that water back out and doesn't let that cell burst. And that's kind of what the cell wall is doing. It's providing a pressure pushing on that cell so that if water is overfilling, the water gets pushed back out. So a plant that's in a hypotonic solution will swell. It'll fill its vacuole. I'm sorry, it took me a minute to think of my word. It'll fill its vacuole with water and be nice and turgid. So you can water a plant with pure water and it'll be fine. It'll take up that water and stand up tall and be a beautiful flower. You'll have a gorgeous, nice yard. In an isotonic solution, your cell will be a little bit flaccid. So that osmotic pressure of the cell membrane, or the pressure of the cell membrane, will push out a little bit more water. And it'll get a little bit flaccid. In a hypertonic solution, do we discuss it here? In a hypertonic solution, there's going to be so much salt and water the cell wall isn't going to stop things from moving out. It only prevents from moving in. That pressure, it doesn't pull anything in. So that water is still going to move out in a hypertonic solution. And that cell is going to start to wilt. And eventually, if it keeps going, that cell membrane is going to shrivel so much that it pulls away from the cell wall. You can see it's actually detaching from the cell wall here. That's called plasmolysis. So a plasmolyzed cell is one that the cell membrane has shrunk down in away from the cell membrane. This is very dangerous. That plant cell is going to die because it can't function like that. So here's plasmolysis. So right here you can see cell wall, the cell membranes are full around it. We're going to gradually see the white is kind of liquid, and then the green is showing you parts of that elodea, this plant cell. You can actually see it shrink in instead of being nice big full squares. So here you can see things are moving around the cells. And see, look here, that cell membrane is pulling in, this cell membrane is pulling in, this cell membrane is pulling in. So all those cell membranes, they're losing their water. So are these in a hypertonic or hypotonic environment? You can go back aside and check and see where does plasmolysis occur. But these cells, look at how this, they're shriveling down their cell membranes instead of filling out their whole cell wall. They're plasmolyzing. All the water is rushing out of the cell, following the salt or following the sugar that's around it, because there's more salt and sugar around it than there is inside of the cell. And that is a result of osmosis. That doesn't take any energy. That's just what cells do when there's not enough salt and sugar inside the cell compared to what's outside. Here is a turgid cell. So is this going to be hypertonic or hypotonic? So you can see these cells are so full. They're a little bit thick. They're turgid. They're swollen together to be a nice strong. That plant is going to stand up nicely. Well, elodea is not going to, but if that were a flower, it would stand up nice and tall. Okay, so can you see the difference between this cell, even if it's a little blurry, the difference between this cell and the cell we just looked at? So the difference between a turgid cell and a plasmolyzed cell. Okay, so next let's talk about facilitated diffusion. Facilitated diffusion is going to use some sort of channel or carrier to let things cross the membrane. So these are things that can't just go straight through the membrane, but it's still going to be passive. It still is not going to take any ATP or any energy, because things are moving down the concentration gradient. So you have a lot of something outside of the cell, not a lot in. It's able to just kind of move in through this channel. A lot of it outside, not as much in, is able to move in through this carrier. Okay, and again, if you remember from that first part of this lecture, channel proteins are just an open pore that anything can fit will go through. A carrier protein it has to come in and make only this specific shape, that specific size. It has to be very specific to fit there, and when it comes in, the carrier protein switches its shape to let it come into the cell. Channel proteins provide corridors, and aquaporins are one type of channel protein. It lets water go through. Ion channels can be specific for ions, so you have to have a specific charge to come through, and only things with that specific charge come through. Some ion channels are called gated channels, and they'll open or close in response to a stimulus and then allow the ions to go through. So in nerve cells are cells that run through your body to send signals from your brain through your body. Your nerve cells will have ion channels that open in response to an electrical stimulus. So your brain actually sends an electrical stimulus that then opens the ion channel. The ions can travel through and your body will move, or you'll do whatever it is your brain is telling you to do. Okay, So everything we've talked about so far is passive transport. It hasn't taken energy. It moves from high concentration to low. There's a different type of transport called active transport. Active transport is when you desperately need to get something in, even if there's not a lot of it around you, or you desperately need to get something out, even if there's already a lot of it out. You're going to move it against the concentration gradient. And your cell is going to have to use ATP to move it. ATP is how your cell stores energy. All proteins in active transport are going to be carrier proteins. Your cell is not going to just non-specifically allow things to move around to take energy. It's going to be very specific carrier proteins that will allow molecules to move and burn energy. So here's a sodium channel. It's actually a sodium-potassium channel. You're going to put your sodium in it. It fills up with sodium. And then it will burn ATP. So it'll take ATP and break it up and use that energy to change shape and allow those sodium ions to go out. Active transport allows cells to keep concentration gradients different from their surroundings. So it is not just everything diffusing and being equal at equilibrium. So one example would be an animal cell has more potassium and less sodium inside the cell compared to its surroundings because it is constantly pumping sodium out and pumping potassium back in. So this is controlled by the sodium-potassium pump, which is this transport protein. It's going to pump sodium out, and then if we had the rest of it, you'd see potassium coming back in, all because of ATP. I'm not actually going to care if you understand quite how the sodium-potassium pump works, as long as you understand its active transport and it burns ATP. So I'm not going to go through this slide. Just know active transport burns ATP.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay, so at the end, you don't have an equilibrium, do you? You have a lot of sodium outside, a lot of potassium inside. It's not an equilibrium. That's what we use that energy for, was to move it against its equilibrium, against its concentration gradient. Okay, so passive transport, we had diffusion. That just straight through the membrane, didn't have any problems. So hydrocarbons, small molecules like O2 or CO2. Facilitated diffusion, that either was just, if it fits, it ships, goes right through it, or certain carrier proteins that have to have a specific shape or charge. And then active transport, that moves things against their concentration gradient and requires ATP. No ATP for any passive. ATP is only for active. Okay, now we're going to talk about a little bit more types of membrane transport, some of the more fun stuff. Play, please.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient. from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient.
We haven't talked about these yet. That's what we're about to do.
Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, so there's one type of transport called co-transport, if you're paying attention to that video. Co-transport is when active transport of a solute will also drive the transport of other substances. So the two things are coupled to each other. So you're diffusing something down its concentration gradient, coupled by actively transporting something else against its concentration gradient. So you can move something in. This is moving in against, like down its concentration gradient, and able to power this moving in, and this can move out against its concentration gradient. Small molecules in water can enter or leave the cell through the lipid bilayer through transport proteins, but large molecules can't typically fit through those transport proteins. They can't cross through the membrane. So they're going to cross in vesicles. So they're going to cross in bulk. And this is going to take energy. Two main types are endocytosis and exocytosis. Endo, so that endo tells you it's moving in, and exo tells you it's exiting, it's moving out. So endocytosis is bringing things into the cell, and exocytosis is moving things out of the cell. We already talked about exocytosis some when we talked about how the Golgi apparatus will prepare vesicles that then take things out of the cell. That's exocytosis. So you already know about that. Let's watch this little video. I think exocytosis and endocytosis really are the most interesting part of the chapter that we're covering this week.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
Okay, so just like that Golgi apparatus making the vesicles fuse out, that's endocytosis. You already know that one. So exocytosis is exiting. I just said that backwards. So the vesicles fusing out is exocytosis that you already know about. Okay, exocytosis is using transport vesicles that are going to move towards a membrane, fuse with it, and release their contents outside the cell, just like you just saw. Some kinds of cells are secretory cells. They secrete things, and they use exocytosis to export their products. So let's watch this exocytosis a little bit more. So here's a vesicle. Maybe it just came from the Golgi apparatus, or maybe it's a particular type of cell that has made some sort of vesicle that is going to be releasing these things out of the cell.
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
Okay. Endocytosis is bringing things in. So you're taking in big things, big molecules, or just stuff around you, but still using vesicles. So endocytosis really is a reversal of exocytosis, but it's going to have different proteins. And there's 3 different types you're going to be talking about. One is phagocytosis. So phago means eating, cyto means cell. So this is cellular eating. Like in this picture, you're bringing something in, and that is going to be food for the cell to eat. One is pinocytosis. Pino means drinking, and cyto means cell. So it's cell drinking. So it's just bringing the fluid around it into the cell. And then receptor-mediated endocytosis. This is going to be when you have receptors that are going to be filled with some, whatever the receptor binds to. And when the receptor binds to its substrate, it just brings it in a vesicle. So in phagocytosis, this is cell eating. This is just a fun word to say. Like, just stop and say phagocytosis. It's a fun word. In this, a cell is going to engulf a particle in a vacuole. So here you have this little amoeba cell that is about to eat this whole little elephant of an algae. It's going to make these, you can see it's starting to make these pseudopods. The pseudopods are going to wrap around and eventually just hug it and bring it in. So this algae is going to be brought into the cell, and that pseudopod is going to, that amoeba is going to feast on it. So you have these pseudopod, encircle it, and bring it in. And then this is going to fuse with a lysosome. If you remember lysosomes, they have enzymes that can digest and break things down. So when this fuses with the lysosome, this food particle will be broken down by lysosomal enzymes down into sugars that the cell can digest for energy.
In endocytosis, membranes invaginate, or pinch in, to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
Okay. Next is pinocytosis. Pinocytosis is just going to be taking up the fluid around you. It does a very similar thing. You form a little invagination, and then it's going to eventually just close off by these two pinching together, and you have this little vesicle filled with fluid.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
Okay. And then last is receptor-mediated endocytosis. And this is not a cell needing food. This is not a cell needing liquid. This is a cell that has receptors that bind to something. And when those receptors are bound, it triggers the cell to invaginate and form this coated pit that has whatever receptor it's bound to. Inside of the vesicle, you'll have the receptor proteins, receptors, all the fluid that's around it. It's all going to be making its way into there. Here's an actual micrograph of this process going on. So you can see this is the plasma membrane. Here's the coat proteins along here, and it's forming in here. You can see it's almost formed this vesicle there. Oh, and then this will empty into, come into the cell. Your cell will do what it needs with it. And then these receptors can go back to the plasma membrane. They can be recycled back.
Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
Okay. So, at this point, you should be comfortable telling me all about osmosis. What is hypertonic, isotonic, and hypertonic. You should be comfortable telling me about active transport compared to passive transport. You should be able to tell me what endocytosis is, what exocytosis is. You should be able to describe phagocytosis, pinocytosis, and receptor-mediated endocytosis. So those are the things we covered. Please make sure you're very comfortable with all of those terms. A lot of this is vocabulary that you just need to flashcard and memorize. But understanding the concepts and the pictures, I think, helps understand those things. And if you have any questions, you can always email me or just talk to me during office hours or after class. And I'll see you soon. We're about to start our metabolism chapters, which are going to be a little intense, but lots of fun too. Okay, see you later. Bye.